so it's already there and you don't have to worry about it. These are both due the day of lecture exam 2, which is October 24th for this class. So by midnight that night. But take pictures and upload, or scan as a PDF and upload the images. That's how you'll submit homework packet 2. I don't want to collect any paper this time, so if you do print it, work with it by hand. Just take pictures of your answers and get it all uploaded into Canvas.
Were we supposed to submit like this first homework packet to Canvas, or?
Either way, you could have submitted it in person or you could have uploaded to Canvas.
Okay.
Yeah, either way. I will give you a warning. So, Unit 2 for this class is a little bit different than Unit 1. Unit 2 lecture is really long. We don't have our next exam until October 24th. That's over a month away, right? So we are a ways out from that. There's a lot of lectures here. The nervous system lectures are a lot. The nervous system itself is a lot. And I'll tell you, by the time we get to mid-October and we're working through the autonomic nervous system, it's going to have felt like we learned muscles forever ago. So the one thing I can say about Unit 2 lecture is to constantly go back and review so that you're feeling like you're managing all of it at the same time. Because like I said, by the time we get to this later stuff, you're going to have kind of forgotten about some of this muscle tissue material, a little bit of the contraction stuff that we're going to go through. We'll have opportunity to review before the exam, the lecture exam, but I just want to put that warning out there. It's a long unit, and you definitely want to just kind of keep constantly reviewing. Go back through your notes from time to time, go back and review. Lab, on the other hand, is opposite that. So lab unit 2 is really quick. Your lab exam is on the 12th. So that's coming up in just a couple of weeks here. We have about 3 weeks, 2.5 weeks of lab before the lab exam. And the lab exam is just going to be on muscles only. So we have muscle histology to look at, and then we'll have the muscles that we're dissecting in our cats. We'll have muscles on our human models. That's it. The muscles hit list, when you take a look at them, they're posted, everything is there for you. The muscles hit list include the names of the muscles and their attachment points. So the bony landmarks that you just learned in Unit 1, you're going to see all those bony landmarks again. The iliac crest, the ischial spine, the acromion process of the scapula, the coracoid process of the scapula. You're going to see all of those bony landmarks again because they serve as attachment points for muscles. Right? When muscles contract, they pull on those bones to generate movement at the joints. So we're going to be learning the names of muscles just to visually identify them. We're also going to learn their attachment points and the actions of that muscle, what happens to the joint when that muscle contracts. That's going to be all for the next couple of weeks here, again, as we're dissecting our cats, as we're looking at human models, and as we're taking a look at that histology stuff. And it's going to go really fast, right, this lab. Again, lab exam 2 is going to come up on Thursday the 12th for you guys. So it's going to almost feel like we're taking 2 different classes, right? And we're going to be focusing on those muscles. Meanwhile, we've only going to spend a couple of days on muscle lecture-wise, and we're moving into the nervous system for the rest of this. So brain, spinal cord, autonomic, we're going to move into all the rest of that. So you're going to be doing muscles in lab and nervous system in lecture. And that's going to be the case for a couple of, about a week and a half or so. And then eventually it'll kind of come back together. You'll see that the lectures and labs eventually catch up with our nervous system labs kind of catching back up here at the end. But just putting it out there to be prepared for, kind of feeling like you're taking 2 different classes at the same time. And just to do your best, I would say, focusing on labs, since that's the first upcoming exam, focusing on lab is probably what I would prioritize, but don't let those lectures of students that are still taking a makeup exam, tomorrow actually. So I can post your scores, and I can give you your Scantron back to look at today, but then I'm gonna collect them again. That works. That way you can at least see what you got, you can see how you did. We'll go ahead and do that after the lecture. Yeah, we'll do that. Lab exams have not been graded. I don't anticipate getting those back to you until Tuesday. So be prepared for lab exams, scores to pop up, and the graded exams to be ready for you on Tuesday. So my goal with all exams is to try to get them back to you within a week. Sometimes I'm successful with that, and sometimes I'm not. But so far, I'm working on the lecture exams, and I got yours done first, so I'm on track. So let's move into Unit 2, and then we'll go ahead and deal with that during break today. Questions? Yeah.
I was gonna say, for these, do you recommend like watching your recorded ones?
You know, I always think that the more you are exposed to something, the better. Maybe even a second time around, it helps things sink in. Maybe after coming to class and listening to a lecture, going back and rewatching the recordings, even at like maybe a slightly faster speed to get through it quicker, you're always gonna pick up on things that you missed. And it just reinforces it. So if you have the time to do that, I probably would. Maybe have my notes in front of me and do it in an active way. Yeah. But it's not required. Certainly students have been successful in the class well before having those recordings available to them. Okay, so we're starting our muscular system. So muscles, their main job is movement. Muscle tissue is the only tissue in the body that moves. So we're going to be talking about muscle tissue today. The skeletal muscle that we're going to be focusing on for the majority of this unit attaches to bones, and so that's what's achieving movement of your skeleton. But there are lots of muscles in your body, like muscles in the wall of a blood vessel that cause vasoconstriction and vasodilation. Muscles in the iris of your eye that cause your pupil to constrict and dilate. Muscles in your stomach wall that cause the churning action. Muscles in your heart, they cause that heartbeat. So movement is really the major function of muscle. But a lot of muscles in our bodies, particularly all those skeletal muscles, are going to have some additional functions. So we get some posture and joint stability here. We talked about how muscles are the #1 stabilizers of joints. Muscles generate heat every time they contract. A little bit of energy is exchanged. ATP is used and converted to energy of movement. And so anytime energy changes from one form to another, some energy is going to be lost as heat. So that's why, like when we're really cold, we start shivering, our muscles are contracting to generate heat. We communicate using muscles, we control our orifices, and muscles can also provide some support and protection. Your abdominal muscles are really the only thing that are really protecting your abdominal cavity, visceral organs from the anterior side. So different types of support there. Muscle. So muscle tissue, all three kinds. We're going to talk today about skeletal muscle tissue, cardiac muscle tissue, and smooth muscle tissue. All muscle tissue have these 5 properties. They are excitable. To be excitable means that you can electrically respond to something, to some kind of stimulus. So there's an electrical event that occurs in the muscle. We'll touch on that a little bit. I know that's a little more physiology. You'll get to a lot of that in your physiology class in the future, but we're going to expose you to that here in anatomy. What about the cell makes it excitable? What kinds of molecules or membrane proteins do these muscle cells have, and how do those work? Muscles are conductive. That means that the electrical excitation can spread. It might start in one location, but it doesn't just stay there. That little bit of electrical excitation spreads down the length of a cell. Or in the case of cardiac muscle, it spreads from cell to cell to cell to cell to cell, so that your whole heart contracts as a single unit. So that's conductivity, is the spread of the Of course, contraction, right? Contractility is a property. That's the ability to shorten. And muscles can also stretch. So they can shorten, they can contract. They can return back to their normal size, regular length, but they can also stretch beyond that. So that stretch is the ability to be extensible. And then anything that stretches in your body, we don't want it to stretch out. We want it to recoil back to its original size, and that's this property of elasticity. So muscles have all 5 of those properties. We're going to talk about the 3 different types of muscle tissue. So just like epithelial tissues, connective tissues, nervous tissues, muscle tissues is kind of a big broad category. And within that big broad category, we have 3 little branches, 3 specific types of muscle tissue. So we'll start with skeletal muscle tissue first. Skeletal muscle tissue has cells that are long cylinders, really tiny cylinders. So we call these fibers or myofibers. So I'm going to draw a long cylinder on the board to represent one skeletal muscle cell here. Okay, that one cell is called a myofiber. And a couple properties about skeletal muscle tissue. One is that these cells are striated. When you look at skeletal muscle cells under the microscope, you're going to notice that the cell looks like it's got these lines running through it. It's going to be a series of dark pink, light pink, dark pink, light pink, dark pink, light pink, dark pink, light pink. So I'm just going to draw a few of these, right, for the sake of speed, but the entire cell would be striated. And I know it looks like it's red and white here, but it's dark pink, light pink, dark pink, light pink. And we'll talk a little bit more about the organization of proteins inside of the cell cytoplasm that create this pattern of dark and light pink bands. But that's what it means to be striated, right? It's to have this banding on the surface. It's visible on the surface of the cell. Muscle cells, myofibers, skeletal muscle myofibers, I should say, are multinucleated. So again, nuclei tend to stain darker, like purple or super dark pink under the microscope. So we're going to see multiple nuclei, but they're just beneath the plasma membrane of a myofiber. And they're not necessarily evenly spaced the way that I illustrated here. They could be side by side. We're just going to find multiple nuclei along the length of a myofiber. So it's a multinucleated cell. This can occur, you know, if a cell undergoes mitosis and fails to complete cytokinesis, right? And you end up with these multinucleated cells. So all skeletal muscle myofibers are striated. All skeletal muscle myofibers are multinucleated. The next property is that skeletal muscle myofibers are voluntary.
So what was the purple line you indicated for the myofiber?
The purple is a nucleus,
Okay.
multinucleated. Yeah, so multiple nuclei per cell in your cell. Skeletal muscle myofibers, this tissue is voluntary. What does voluntary mean? You ready?
You have control over it.
Yeah, you've got conscious control over the ability to contract these cells. So when, how strong, with which force, that's all voluntary. That means it's controlled by your conscious nervous system. And then the last thing to talk about for skeletal muscle is where we find this tissue. We find skeletal muscle in all of our voluntary muscles throughout our bodies. These are most of the muscles that are going to be attached to bones, moving your skeleton, but we also have lots of skeletal muscles in our face. Those facial muscles, they're actually attached to your skin, right, moving the skin. The only movable bone in your head is your mandible, right? Everything else is fixed. So your facial muscles, they're all skeletal muscle as well, but they're moving the skin in a voluntary way. Okay, next up is cardiac muscle. So cardiac muscles are second tissue here. Cardiac muscle cells, they're called cardiocytes. They have a different shape. They're kind of branched, so you'll see them, they look a little bit more stumpy, and they're shaped like maybe a letter Y or maybe like a letter K. cells, or cardiocytes. So one cell is a cardiocyte, and the tissue is referred to as cardiac muscle. Skeletal muscle is probably making that tissue. So let's talk a little bit about cardiocytes and their properties. Cardiocytes are usually uninucleated, one nucleus per cell. Occasionally, you might find a cardiocyte that has two nuclei, but never any more than that. The other key about the nucleus in a cardiocyte is it tends to be big and round. So you'll find a big, round, dark nucleus right in the center of the cell when you look at these guys under the microscope. So we'll do one nice big nucleus for each cell here. Again, sometimes you might find two nuclei, but usually just one. Cardiocyte cytoplasm is going to look striated as well. So again, our proteins are what's stained pink under the microscope. Anytime you see the dark and light pink bands, you gotta be thinking something's going on with the proteins here, right? Because that's what I'm seeing, are these proteins that are staining pink. So same thing here with the cardiocytes, we're gonna see striations, dark pink, light pink, in each of these cells. I'm not going to draw for all of them, but all the cells would be striated. Cardiocytes are, or cardiac muscle tissue, I should say, is an involuntary tissue. So we don't have conscious control over cardiac muscle tissue, right? I don't have to think about my heart contracting. It's involuntary. It is controlled by our nervous system, but the involuntary side of our nervous system, which is known as the autonomic nervous system. We'll be talking about that much later in Unit 2. And then the last thing is, where do we find cardiocytes, or cardiac muscle tissue? There's only one organ that contains cardiac muscle tissue. That is the heart. The heart. That's it. So the only organ in your body that contains cardiac muscle tissue is your heart. The last tissue type is smooth muscle, and there are a couple of smooth muscle cells there on the screen. Smooth muscle cells tend to have this, they call it a fusiform shape, where it's a little thicker in the middle and tapers on either side. Fusiform or spindle-shaped cells. So when you see smooth muscle, you're going to see these cells kind of tapering. They tend to kind of pack together long ways. Something like that. Just a group of smooth muscle cells. These smooth muscle cells have one nucleus per cell. They're called uninucleated, just like our cardiac muscle. Unlike the case with cardiac muscle, the smooth muscle cells are definitely uninucleated, never two. I'm sorry, I know this thing's in the way, I wish I could move it. Feel free to move around if you need to. So one nucleus per cell, and the trick here with smooth muscle is the nucleus is kind of long and flat, very much similar to the shape of the cell. So you get these long, flat nuclei in each cell. Again, just one per cell. Uninucleated. Smooth muscle tissue is non-striated tissue. So the cytoplasm still has a whole bunch of proteins in it, but they're not arranged in those lines, in those banded patterns. The proteins are kind of universally dispersed in a smooth muscle cell. So the cytoplasm will still stain pink, but it will stain fairly uniformly pink, as opposed to striations, right? You just see this uniformly pink cytoplasm with a purple nucleus. Again, every cell would look like that. So non-striated, uninucleated is the characteristic you'll look for smooth muscle. Down here. So tissue type is called smooth muscle. There's no special name for the cells, at least none that I'm aware of. We just call them smooth muscle cells. Smooth muscle tissue is involuntary. Again, you don't have conscious control over this tissue, just like cardiac muscle. The involuntary tissue of smooth muscle will move organs in your body then that move without your conscious control. Anything in your body that's not the heart, that's moving without conscious control, is going to be moving because of smooth muscle. So can you name a couple of organs in your body or a couple of things that move in your body? What is it?
Your intestines.
Your intestines move. Yeah, your intestines are churning and moving the food through. Yeah, that's a great example. So there's smooth muscle in the wall of your small and large intestines.
Erector pili.
Erector pili. I love it. Thank you. Yes. Where's the erector pili muscle?
In the skin.
In your skin. Yeah, so that's a great one. That's one we've already learned about, right? Causes goosebumps. What else has smooth muscle, do you think? What happens when you shine a light in your eye? Yeah, pupils constricting and dilating. Your iris, right? That colored part of your eye, that's actually a smooth muscle ring. It has some pigment on its anterior surface, and that's what we can see as like somebody having brown eyes or blue eyes or green eyes. But it's actually a ring of smooth muscle that's controlling the size of your pupil. Blood vessels, right? Anybody like skin turns bright red when they get hot? Blood vessels are dilating. Whereas if your skin turns blue when you're cold, blood vessels are constricting. That's all movement generated by smooth muscle. So cardiac muscle is in the heart. Smooth muscle is in every other organ that moves in an involuntary way. Yeah. The air passageways in your lungs, your bronchi. Anybody here have asthma? Right? That's your smooth muscle, right? Wrapped around those organs. All those other organs. Quick note here. I'm just going to say it. On a test, if I were to ask you to tell me the muscle type that's found in a blood vessel that's attached to the heart, like the pulmonary artery, or the aorta, or the superior vena cava, or the inferior vena cava, right? We're talking about a blood vessel attached to the heart. What tissue type do you think is going to be found in these blood vessel walls? Would it be cardiac muscle or would it be smooth muscle? Smooth muscle, right? Because cardiac muscle is only found in the heart itself, not the vessels, not the arteries, not the veins, right? Smooth muscle would be found in your arteries and veins. And of course, arteries and veins attach to the heart, but they are not the heart. They are not part of the heart. Just putting it out. Okay. So, a couple of examples here. This is what you'll be looking at under the microscope. We're not using the scopes today. We'll do this on Tuesday of next week. Today we want to get through our cat skinning. But next week you'll want to bring some of these pictures with you or pull them up on a device so that you know what you're looking at. But here's where our cardiac muscle cells, our cardiac muscle tissue looks like. Cardiocytes, so this would be an example of one of those Y-shaped cardiocytes here, right? Nice round nucleus, and hopefully you can appreciate that there are these little striations in the cytoplasm of the cell. It kind of looks like it's a little bit striped there or banded. If you look up really close, you can probably see that a little better. One thing to note about cardiocytes is that the cells are connected to one another through special junctions. There are these special junctions between cells called intercalated discs, and oftentimes those intercalated discs are obvious under the microscope as well. And that's what the blue arrow is pointing to, right? A special junction between neighboring cells. And again, it's called an intercalated disc. Those would be found between all cardiocytes. This is what allows one cardiocyte's electrical excitation to conduct to its neighbor, to its neighbor, to its neighbor, so that that electrical excitation spreads uniformly throughout cardiac muscle tissue. does not happen in the same way with our other muscle tissue types. Here's smooth muscle. So smooth muscle to me looks a lot like dense regular connective tissue. When I look at it under the microscope, it always takes me a minute to figure out, am I looking at smooth muscle or am I looking at dense regular connective tissue? One of the things that I look for is the positioning of the nuclei. Fibroblast nuclei, you know, also look kind of flat like this. So when you see them in dense regular connective tissue, you're going to see these flat purple nuclei there too. The difference is the pattern of them. In smooth muscle, the nuclei are kind of randomly distributed. They seem like they could be anywhere. They also, if you look really carefully, they are surrounded by pink cytoplasm, right? Completely. You can kind of make out the shape of that cell there and see that the nucleus is inside of that pink cytoplasm. That the pink that you're seeing here is not collagen, right? Which is what it is when you're looking at dense regular. It's collagen outside of the cells. In this case, the pink is the proteins that you're seeing in the cytoplasm of the cells. These are all cells that are staining pink here, no collagen. And then when you're looking at dense regular connective tissue, you see all these like patterns of collagen, like the pink kind of all by itself, and then you'll find the fibroblasts in rows, right, that are kind of between those pillars of collagen. Versus again, smooth muscle, where the nuclei seem to be anywhere and everywhere. They're not in rows, they're not in a pattern, they're not arranged, lined up like kindergartners, right, like they are in dense regular. So maybe when we get to the histology, I'll remind you about this on Tuesday, but maybe just take a second look at your pictures of dense regular and compare that to what you're seeing under the microscope for smooth muscle. Okay. And then skeletal muscle. So here's what we're seeing for skeletal muscle. The picture on the left is showing us 4 myofibers. You've got 2 nice big ones in the middle, and then a third and a fourth just at the cutoff at the top and not at the bottom of the image. You can appreciate the shape of the cell, right? This is kind of a side view or longitudinal view of the cell with that cylinder shape. You can see the light pink and dark pink bands pretty nicely. So you see the cells are striated, multiple nuclei per cell. Right? That's what we're seeing here with these dark purple spots. The nuclei are always just beneath the plasma membrane. The majority of the cell cytoplasm is just chock full of all of these proteins, and these dark pink and light pink bands, right, crossed by those proteins, these are the proteins that are going to contract or generate contraction. When we look at skeletal muscle in a cross section, so you're going to see longitudinal view or side view, you're also going to see skeletal muscle that's been cut in a cross section. So imagine having a whole bunch of these cells in a tissue and instead cutting them this way, right? You're no longer going to see the striations. Of course, the tissue is always striated, right? So this is always going to be considered striated tissue. But I no longer see the striations because I've cut through each of those bands, right? The darker light pink bands. So it's not going to be obvious. You're just going to see a pink cytoplasm, right, full of those proteins. In this cross-sectional view of these myofibers, you can also appreciate that the cells are multinucleated. So I can see some areas where there's a couple of nuclei in the same cell. This guy has 2 nuclei in the same cell. But that's not always going to be apparent in a cross-section either, right? Just imagine cutting a cross-section through this cell here. I might cut a cross-section through an area of the cell where there's no nucleus. I might cut a cross-section through an area of the cell where there's only 1 nucleus, right? So it's not always going to appear that every single cell has multiple nuclei in a cross-sectional view. Again, it's always considered multinucleated tissue, multinucleated cells. Whether I can see it or not in my particular section doesn't matter, right? Skeletal muscle is multinucleated. Skeletal muscle is striated. Question?
When you label muscle tissue, do you have to say like striated in the beginning like you would like ciliated?
No. It's just a characteristic of the tissue.
Okay.
Yeah, it's not part of the tissue's name.
Okay.
here, right, just to show you a comparison side by side. This is, I think, somewhere in the face. I feel like this is probably a facial muscle because this looks like a salivary gland to me. So I think we've got muscle, right? What's here? What's this white tissue?
Adipose?
Adipose, right? Muscle and adipose side by side. So muscle kind of resembles adipose, except that, remember, adipocytes, the cytoplasm is empty, because it's filled with that lipid vacuole that gets washed away during the preparation process. So you end up with these, plasma membranes with empty space inside. Versus a cross section through myofibers, right? You can actually see it that there's pink proteins filling the cytoplasm. All right. So we're not going to spend too much time in this unit talking about cardiac muscle and smooth muscle beyond the general characteristics that we've already mentioned. So we'll see cardiac muscle again, and we'll focus on it again when we get to the cardiovascular system. We'll see smooth muscle again. There's not a whole lot more to say about it, but we will see it again when we get to all of our other organ systems. So blood vessels in the cardiovascular system, the iris of the eye, the digestive tract organs, the respiratory tract organs. Right? We'll see smooth muscle in all those organ systems that it pops up again, or pops up in. But we do spend quite a bit of time in this unit 2, focusing on skeletal muscle. So that's going to be kind of the continued conversation about muscle, is with skeletal muscle as our primary example. So you can appreciate here that we've got a myofiber in that top picture. Myofibers are long cylinders. They are multinucleated cells, so all the purple spots are the nuclei, and they're striated, right? They have that dark pink and light pink banding pattern. But what we haven't seen yet, what we're going to get into, is what's found in the cytoplasm. What are the structures that are causing those striations in the cytoplasm of these cells? And those structures you can see here in this illustration are actually small little kind of mini cylinders, right, inside of the larger cylinder. Those mini cylinders are called myofibrils. Myofibrils fill up the cytoplasm of our myofibers. Okay? So this picture here is taking a look at one single myofibril, just one of these guys, right, blown up. One single myofibril is actually made up of small little kind of strings or small little rod-like proteins. And those small little rod-like proteins, the yellow ones and the red ones, you can kind of see how they're alternating here. Those guys are called myofilaments. A lot of myo words. Myo is our muscle root word, if you haven't figured it out by now. Right? So we've got a myofiber, that's the name of the cell. The cytoplasm of the myofiber is full of this organelle called myofibrils. Myofibrils, we call these the contractile organelles of a myofiber. These are the things that will contract. Myofibrils, again, they're the contractile organelles of a myofiber. And so we could find a myofiber with hundreds of these loaded in the cytoplasm. What's a myofibril made of? Myofibrils are made of these little rod-like proteins called myofilaments. So the red and the yellow proteins, those are the myofilaments. Here we're looking at a really close-up view, like super zoomed-in view of the myofilaments. And you can see here that the myofilaments, one of them is thick and one of them is thin. The thick and thin filaments, they overlap one another. What they've shown in the illustration is two different colors here. So the gold filament is the thick one and the red filament is the thin one. But remember, these are just proteins. So when we stain using our pink and purple combination, the H&E stain, this is all going to stain the same color. Right? The thick filaments are going to stain pink, the thin filaments are going to stain pink. So this pattern of alternating rows, they stack up on each other. So thick filament, thin filaments. Thick filament, thin filaments. And then you're gonna have another row of thick filaments and thin filaments. So if this is all staining pink, you can imagine that that's gonna give you an area where you have dark pink, light pink, dark pink, light pink. That's what causes the striations. Okay, so we're gonna come back to all this and we'll go through this in a lot of detail. We'll take a close look at the proteins that these filaments are made of. But that's what's contributing to the striations, this organization of the thick and thin filaments within myofibrils that are stacked and loaded inside of a myofiber. Okay, we'll see it again. Promise. So skeletal muscle, cardiac muscle, and smooth muscle. Those are our three muscle types. You've got examples of each of the types here, right? Some different pictures that you can look at, pull up some images online. You'll see they look pretty distinct, each of these muscle types. These three muscles, and we'll see some dense regular connective tissue. These guys are going to be what you're going to be looking at on Tuesday, right, for lab on Tuesday, part of your lab. The table here kind of helps organize like some of the major characteristics of these tissues. So one of the things you want to know is where do we find that tissue in the body? Okay, so have an example of where we find each of these tissue types in the body. We'll talk about the connective tissue components in a moment, so we'll skip that one for now. We'll skip the calcium source. Myofibrils. The presence of myofibrils here, this column, refers to whether or not the tissue is striated, right? So it's the myofibrils, the organization of those thick and thin filaments that create the striated patterns that we can see under the microscope. So that should tell you, myofibers, yes, there are myofibrils in myofibers. Cardiocytes, yes, there are myofibrils in cardiocytes, right? That's what's creating the striations in our cardiac muscle tissue. Smooth muscle, no myofibrils. We're going to still have contractile proteins, but they're not organized in cylinders. They're not organized in alternating rows or patterns. So smooth muscle is non-striated because there are no myofibrils. Okay, so this visual characteristic that we can see is due to the internal anatomy of the cell. Okay, what's going on in that cell? So that's what I wanted to point out here for a comparison of each of these tissue types. So continuing on with our muscle words today, right, a lot of these words that we're going to be incorporating into our anatomy of a myofiber, anatomy of a cardiocyte, they're going to include these roots myo and sarco. So myo means muscle, sarco means flesh. And that's where a lot of the words are derived from. Muscle, the word itself, is actually related to the genus and species name for mouse. So mouse genus and species is Mus musculus. Muscle is actually derived from this mouse idea because it used to kind of appear, created this idea in somebody's head, whoever just, I don't know who named muscle. But the story is that somebody thought that the biceps brachii muscle looked like a little mouse running in somebody's arm. And so that's where the name muscle came from. Right? Mus musculus, little mouse in your body. Right? Pictures of bodybuilders, you can see muscles can get really big, muscles can be really small. What's the difference between somebody who has large muscles and somebody who has average size muscles? It's actually not that this person, Schwarzenegger here, has more muscle cells, right? So myofibers, that's what's making up this biceps brachii muscle here. Each individual cell in his biceps brachii is larger, right? Is bigger. So muscles don't grow in your body by cell division. Muscles grow by what's called hypertrophy. That means that each individual cell gets larger. What happens is inside of this myofiber, we know it's loaded with all those myofibrils. This cell can get larger by filling up with more myofibrils, right? Getting more contractile proteins, and that's going to generate more force when the cell contracts, right? That's how cells get stronger. And if a muscle organ is made up of many, many cells, right, then each cell getting larger is what's making that whole organ stronger and bigger. Question?
I was gonna say, isn't that like kind of like adipose, but I guess it doesn't like add fillers, but like everyone has the same amount, right?
Kinda, yeah, it's a good analogy, yes. Adipose works the same way, very much. One of the things that's interesting about muscle cells is while they can grow individually like this, and they'll grow when you strain. So that's, somebody goes to the gym, they lift weights or do whatever their daily activity is, they strain their muscle or exert and exhaust the muscle. The muscle then responds by growing, right? Transcription and translation of the genes in the nuclei is what's going to create all the proteins that the cell fills up with. Make more myofibrils inside of that cell. And that's a response to the stress. But basically, your muscles will grow to accommodate the tasks that you charge them with, your everyday activities. If you all of a sudden have a child in your home that you have to pick up and lift, it's going to be difficult at first. Your muscles maybe aren't used to that, but you strain your muscle responds by growing, right? That's how we grow our muscles. So somebody who's a bodybuilder has to strain their muscles to respond by growing. Hypertrophy will occur. On the other hand, if you don't use your muscles, if you don't use these myofibers, you'll lose them. So they will atrophy, which means they get smaller and smaller and smaller, but they will eventually die. And once a myofiber is lost, it's lost forever. You don't ever grow new myofibers. So somebody who's bedridden, somebody who doesn't use their muscles, somebody who's paralyzed, right? The muscles will atrophy. They will be lost, right? And the cells will die eventually over time. All right, so let's take a look now at muscles as organs. Just to give you a little perspective on the picture here, this is a muscle. This is actually one of the heads of the biceps brachii muscle. And I know that because I can see the tendon of this muscle going through the intertubercular sulcus or intertubercular groove of the head of the humerus up there. I'm driving through there, it's going to attach to the scapula crossing the joint. So just to kind of give you the idea, this entire thing here, this is the organ here, right? It's the whole muscle. If we're looking at our models, right? This is the muscle that we're looking at here, this biceps brachii muscle. So each muscle organ is made up of multiple tissues working together for a common purpose. Right? That's the definition of an organ. So let's take a look at the organization of the muscle organ. The tissues here that we find are quite a bit of skeletal muscle tissue, right? That's definitely the majority of the organ. Skeletal muscle tissue loaded with myofibers, right? All throughout this organ. But there's other tissues here. There's connective tissue. Okay? There's this connective tissue wrapping around the outer surface of the organ. And that connective tissue wrapping around the outer surface of the organ is called the epimysium. Right? So myo-mes, right? That's muscle. Epi means like to surround something, the surrounding. So we've got epimysium surrounding the outside of every single muscle organ. Every single one. Surrounded by epimysium. Epimysium is a thin layer of dense, irregular connective tissue. And it is continuous. The collagen that we find here in this epimysium will continue into the tendon. And what tissue type are your tendons made of? Dense, regular connective tissue, right? It's the same ingredients as dense, irregular. The difference is the organization of the collagen, the pattern of collagen. In dense irregular, collagen is kind of running every which way. In dense regular connective tissue, the collagen all lines up and runs parallel to be really strong in that one direction. So epimysium surrounds the outside of your muscle, and it continues then as the tendon that will attach to each bone at each end. If we look inside of each muscle organ, what you're gonna find is the myofibers are not just all clumped together as one giant unit. You're gonna find the myofibers are actually bundled into these little kind of sub-bundles. So this right here is a bundle of maybe 20 myofibers. Here's another bundle of 20 myofibers. Here's a bundle of 12 myofibers. Each bundle of myofibers is called a fascicle. And those fascicles are surrounded by dense irregular connective tissue called perimysium. So epi is on the outside, peri is inside of the muscle, surrounding the fascicles, bundling the cells into fascicles. Perimysium, it's the same material, it's dense irregular connective tissue. Same material as the epi, the difference is in the location. Collagen fibers running every which way, wrapping up the fascicles. If you took a closer look at a single fascicle, so this is a fascicle here, maybe it contains about 25 cells, 25 myofibers. Right? It's wrapped in perimysium on its outside. Each individual cell then is a myofiber, and each myofiber is surrounded, outside of its plasma membrane, it's surrounded by a small amount of areolar connective tissue called the endomysium. So endomysium wraps around an individual cell, an individual myofiber inside of a fascicle. If we were to, yeah?
So the perimysium is like the whole thing around it, and then the endom will be around just one myofiber?
Yes, endo is around each myofiber. If I had, if this were accurate, I would find some endomysium here, right? Areolar connective tissue, endomysium. A bundle of myofibers is wrapped by perimysium. So the entire muscle organ, if we sliced it in cross section, we'd find the epimysium on the outside, and that's again dense irregular connective tissue. So lots of collagen fibers running every which way, like the dermis of your skin, right? Like your particular dermis. It's just a thin layer, right? Not as thick as the dermis. Inside, we're gonna find fascicles. Each fascicle is surrounded by a layer of dense irregular connective tissue called perimysium. So here is my perimysium. Right, so you can see that your perimysium is actually continuous with your epimysium. And it's all based on location. When it's inside, we call it perimysium, and when it's outside, we call it epimysium. But it's the same stuff. Dense irregular connective tissue. So perimysium would be between the fascicles. That's all considered perimysium. Inside of a fascicle, so this is a fascicle, we're gonna have myofibers. Okay, myofibers cut in cross section, remember, look like pink circles that are multinucleated. I'm gonna go ahead and fill these in. This is a cross section of myofibers. And remember, sometimes we're gonna catch multiple nuclei in the cross section. Sometimes we might not catch any nuclei in the cross section. It just depends on the location. Okay, so we'll go ahead and draw that kind of variable here. Some of these guys have multiple nuclei, some have none. So those are my myofibers. And between all of the myofibers is my endometrium. So this area in here. An endometrium is loose areolar connective tissue. This is dense, irregular, and this is areolar. Loose, lots of brown substance surrounding the cytoplasm of those cells. Or the plasma membrane, excuse me, surrounding the plasma membrane of those cells. Let's talk a little bit more practical. So when you look at your cat muscles, or when you look at meat, like, I'm sorry for anybody who might be grossed out by this, but meat, right? That's what we eat when we eat chicken breast or, a nice steak, right? It's the muscle of the animal. So when you're looking at those muscles, you can actually see the epimysium on the surface. If anybody's ever handled like a raw chicken breast, there's like this continuous film on the surface of your chicken. That's the epimysium of that particular muscle, right? It's the pectoralis major muscle of the chicken, right? That's what chicken breast is. So you can see that film kind of defining the outer boundary of that muscle. When you cook your chicken, right? I'm a big fan of the crock pot. I work a lot. We crock pot it up, we shred the meat at the end of the day. My family loves it. We eat tacos every day. It doesn't matter. When you shred your meat, right? What you're doing is you're cooking. So you're applying heat to break down this collagen. Collagen proteins denature, right, with heat. You're denaturing the epimysium and you're denaturing the perimysium. You're breaking down that collagen so that now the perimysium between these fascicles breaks down and becomes shreddable. Each little meat shred that you get is a fascicle, right? It's not a single cell, it's a fascicle. And you can't shred raw meat because that collagen is pretty strong, right? That collagen in your perimysium, that collagen in your epimysium, it's not gonna pull apart, right? It's strong stuff. It has tensile strength. It's resistant to tearing forces. You have to cook it to break the collagen down so that you can then tear it apart. Next time you go to the grocery store, take a look at the steaks in the meat counter, like the nice big ribeye type steaks, cut in cross section. You can see adipose tissue accumulates in the epimysium. Adipose tissue accumulates too in the perimysium, right? But you can see these fascicles, you can visibly see it in those sections. When we're doing our cat dissections, I'm gonna say stuff like, follow the grain of the muscle, like the grain of the meat, right? Tri-tip is a meat that I'm super picky about cutting cross-sectionally, cutting against the grain, because it just makes the pieces a lot more tender. My husband doesn't quite understand that. I'm the meat carver in our family, probably because I dissect cats, I don't know. But cutting against the grain versus cutting with the grain, right? If you've ever talked about that or seen that in your kitchen, right? The grain of the meat is due to the fascicles. You can see fascicles with your naked eye. You can't see individual myofibers, but you can see fascicles. So we're gonna talk about fascicle direction in our cats. We're gonna see the grain of those fascicles. They're etched on all the models, right? These little lines that are etched on the models are the fascicle directions, the pattern of the fascicles, the grain of the muscle. Okay, so those are our connective tissue wrappings. And of course, right, there's more than just connective tissue and muscle tissue in our muscles. There's blood tissue, right? We've got lots of blood vessels running through these. There's nervous tissue. This is all voluntary, so there have to be neurons that are controlling all these cells. So there's a bunch of tissues here in our muscle organs. The top picture, or top terms here, superficial fascia versus deep fascia. So fascia is a term that's given to the connective tissue that's not the outside of the muscle, but kind of the filling stuff in between muscles and other organs. Superficial fascia is the name that's given to the hypodermis. This is the separation between your superficial muscles and your skin. When we clean our cats today, we're going to be skinning and removing the dermis. I'm going to want you to remove as much of that hypodermis as you can as well. We don't want to leave adipose tissue on the surface of our cat's superficial muscles. We want to get that off as much as possible. So that hypodermis in relationship to the muscle is superficial to the muscle, and that's where this name comes from, superficial fascia. So oftentimes, instead of saying hypodermis, when I'm talking about it attached to the muscle, I'll use that term, superficial fascia. Get that superficial fascia out of there. Deep fascia is the connective tissue between muscles that are layered. I can't take this arm apart, but in your body, there are lots of muscles that are like one right next to another. And of course, if muscles are contracting all at the same time, right? Like when I'm moving my wrist, it's due to contraction of muscles in my forearm. And I have more than just these superficial muscles in my forearm. If I remove this guy, you can see that there are muscles deep to these muscles. And if all these muscles are contracting, we don't want them rubbing against one another because that would generate friction. So there's stuff, there's like a filling in between these muscles. That filling is the deep fascia. Deep fascia, similar to superficial fascia, can contain some adipose tissue, but oftentimes it's more of an areolar connective tissue. And what we're going to see when we start removing the superficial and when we start breaking up deep fascia is as you spread it apart, it almost looks like a cobweb to your naked eye. So when we're doing this in the cat, you'll see this like white cobwebby material. Those are the collagen fibers, and when you spread them like that, you can actually break them up really easily with your fingers, a blunt probe. You don't need to cut through them at all. So deep fascia and superficial fascia usually kind of peel off or break apart pretty easily in our cat dissections. All right. So we've got a lot of F words here, right, with muscles. Keep it clean, right? We don't want to go with the angry F words. But here's a slide to kind of help you keep it straight. So again, let's start big. A big muscle organ is full of fascicles, right? A fascicle is a bundle of cells, right? So there are many fascicles surrounded by perimysium inside of each muscle organ. If you looked at a single fascicle, you would find that the fascicle is full of myofibers. A myofiber is one muscle cell, right? One skeletal muscle cell. Each myofiber, remember, is surrounded by endomysium, areolar connective tissue. Inside of a myofiber, it is loaded with tons of tiny little cylinders, the contractile organelles called myofibrils. Right? Myofibrils, the contractile organelles, are filling the cytoplasm of the cell. The myofibrils are responsible for creating the striations that we can see when we stain our cells for histology. Why are the myofibrils creating the striations? Because the myofibrils are made of myofilaments, the thick and thin filaments that are alternating rows to, again, ultimately create this pattern of a dark pink and a light pink banding pattern or striation pattern on the surface of our cells. So myofilaments are those proteins that the myofibrils are made of. Little more vocabulary for today, and then we'll wrap it up, just to kind of finish up on our cell anatomy vocabulary. So this is a slightly different picture of a myofiber. This is one myofiber right here, one cell, just to give it perspective. In this illustration, this myofiber, they've drawn the myofibrils to be pretty large. So there's only, 10 myofibrils inside of this cell, versus the previous example that we saw here, this is the same thing, and this one has maybe 100 myofibrils. So it's just, the artist's rendering of these guys. But one myofiber there is the same as one myofiber here. Okay. Some terms down here to define. So the plasma membrane of a myofiber, this kind of clear wrapping along the outside, that's the plasma membrane. It gets a special name in muscle cells. It's called the sarcolemma. So sarco, right, there's one of our muscle roots. And lemma, if you remember way back in the beginning of class, we talked about the plasma membrane pretty significantly, right? Diffusion of molecules, facilitated diffusion, active transport. We defined terms, and at that time, one of the terms we defined was a plasma lemma. And I quickly said a plasma lemma is a plasma membrane that holds a charge. That's this guy. So because it's the muscle's plasma membrane, we call it the sarcolemma. So the sarcolemma, the plasma membrane of the muscle cell, it's going to hold the charge. Right? We're going to see this electrical charge at the sarcolemma. So that's to be continued in the future. We'll talk about it more on Tuesday. Right now, just know its name, sarcolemma. T-tubules. So T-tubules are invaginations of the sarcolemma that penetrate into the cytoplasm of the cell. On the illustration here, there are these green tubes. You can see that the T-tubules are connected to the sarcolemma. So they are like literally these invaginations of the sarcolemma into the cytoplasm. A continuation of the sarcolemma deep into the cytoplasm of the cell. So all the green tubes that you're seeing are representing the T-tubules. This is going to be relevant again on Tuesday when we talk a little bit about muscle excitation and contraction. We're going to talk about how the excitation starts at the sarcolemma, the surface plasma membrane of the cell, but that excitation has to get transferred into the cell cytoplasm somehow. So that's really where the T-tubules come into play. Sarcoplasm, it's just a muscle-related word for cytoplasm. Right? All of this stuff in here is considered the sarcoplasm. The T-tubules are part of the sarcoplasm. The myofibrils are part of the sarcoplasm. The mitochondria, right? They're all part of the sarcoplasm. It's just the cytoplasm of a muscle cell. Sarcoplasmic reticulum. So this word is a lot like the endoplasmic reticulum, and that's exactly what it is. Sarcoplasmic reticulum is the endoplasmic reticulum of a myofiber, of a muscle cell. And it is the smooth endoplasmic reticulum. It's not rough. So smooth ER of a myofiber is called sarcoplasmic reticulum. If you remember, rough ER was a site of protein synthesis, but smooth ER stores things. And that's what we're talking about here. Just to give you a preview for Tuesday, this smooth ER, the sarcoplasmic reticulum, one of the important things it stores is calcium. And that's going to come into our muscle contraction story.
I'm sorry, so the T-tubules are the closest to what in the cells?
T-tubules are invaginations of the sarcolemma. It's like connected to the surface, like pulling that inward. Okay, so smooth ER stores calcium, sarcoplasmic reticulum. The sarcoplasmic reticulum on the illustration is all of that purple webbing that you're seeing. And if you'll notice, that purple webbing of the smooth ER, or the sarcoplasmic reticulum, it actually wraps around these myofibrils. Right? It's kind of everywhere, all throughout the sarcoplasm of the cell. You find sarcoplasmic reticulum. If you look at the ends of the purple webbing, you'll see that this sarcoplasmic reticulum creates these edges that butt up against a T-tubule. So the purple edge that butts up against a T-tubule is known as a terminal cisterna. Terminal cisterna is the edge of the sarcoplasmic reticulum. together, 2 terminal cisternae, and 1 T-tubule in the middle form what's called a triad. So the T-tubule in the middle, and then the 2 flanking terminal cisternae. Right? Those 3 things form a triad. We're going to see that that triad is used to initiate a muscle contraction. So looking from a myofiber, this is one myofiber. It's got a sarcolemma on the outside. It's multinucleated with the nuclei just deep to the sarcolemma. This one has many, many myofibrils. Right? We're not seeing it, but wrapped in between and around these myofibrils would be the sarcoplasmic reticulum. Right? We have T-tubules invaginating from the sarcolemma. We're going to take a close look at the myofibril to get our last little bit of defining the dark and light bands that we're seeing over here. So it's the myofibrils that are causing the appearance of striations on the surface of the cell. And looking carefully at a myofibril, we're going to break it down. Remember that I drew this earlier image showing dark pink bands and light pink bands caused by the thick and the thin myofilaments. So here are my thick myofilaments, and the way that they arrange in a pattern inside a single myofibril is in rows. And you find multiple rows of these thick myofilaments. So these guys are the thick ones. And then just for the sake of like your own appearance and what you can see on the board, I'm gonna draw the thin ones. I'll just stick with blue. But remember that this is a protein, and so under the microscope, these thin myofilaments, yes, I'm saying the right word. These thin myofilaments, they're gonna look pink under the microscope as well. So they would stain pink, but for my picture, I got a little off there. I'm just gonna draw the blue here. And the thing about these rows of thick myofilaments, thin myofilaments, thick myofilaments, thin myofilaments, is that they slightly overlap. So you can see this small area of overlap on either side of the thick and thin myofilaments. Just a little bit of overlap there. So this whole thing that I just drew would be one giant myofibril. Okay, that whole thing, one giant myofibril. And we're gonna go ahead and label, right? There's areas now. Within this organization of these rod-like proteins, you can kind of see certain patterns, and you can see certain areas appear. Before I begin labeling all these bands and areas, let me just define my thick and thin filaments. These thick filaments are made of a protein called myosin. So I sometimes say myosin thick filaments, but whenever you hear the word thick filament, the protein that makes that thick filament is myosin. The thin filaments are made of a protein called actin. Again, both are proteins, both stain pink with our pink and purple combination of dyes. So this is what's going to give us an area of a band of dark pink and an area of a band of light pink. The dark pink bands that we're seeing on the surface of the cell get a name. They're called the A bands. This black different. This guy. So from like here to here, that's going to be my dark band. Right. dark pink, so it's called the A band. And then wherever I find thin filaments only, right, the band will stain light pink because the thin filaments are thinner, right? Less protein means less pink getting absorbed there. So here, the light pink band is called the I band. So this arrangement of a single myofibril will be repeated for every neighboring myofibril inside of the entire cell. And so when you look at a single cell, a single myofiber, you're going to see a light band and then a dark band, and then a light band and then a dark band on the surface of the cell. So we can define those surface bands as A bands and I bands. The dark band is the A, the light band is the I. But we can break this down all the way into a single myofibril and define our A bands and I bands, like we just did with the single myofibril, super zoomed in on the board right there. Right? A bands and I bands, dark and light. So we're going to define these. What do we find? An A band is located wherever we have the myosin thick filaments. Myosin thick filaments are always found in the A band. But I can't say the same for the I band. I can't say that an I band is wherever we find actin thin filaments. Because there's going to be some actin thin filaments within an A band, right? Because of the overlap. So while the edges of the myosin are defining my A band, I have to say that it's like absence of myosin. Or I can say that the I bands are found where there's actin thin filaments only. Okay, that's going to define my I band. But it would be incorrect to say that I bands are found wherever there's thin filaments. The thin filaments don't define my I band. It's thin filaments only. And that word only needs to be there. Or you could say the absence of myosin thick filaments defines your I band. Does that make sense to everybody? Yep. Absence of myosin or having thin filaments only is what defines the I band. And that's what's going to stain later. Okay, let's go back to the words up there. So next up, we'll go ahead and define the M line. The M line is found in the middle of every A band. I'll just illustrate it here. It's right down the middle, the M line. And it's actually a series of proteins that are kind of binding all those myosin thick filaments together. You don't really need to know what it is, more of where it is. So the M line is located in the middle of those myosin thick filaments. In other words, it's in the middle of the A band. Every single group of myosin thick filaments has an M line. It's next on the list. H zone. The H zone is the area of the A band that contains myosin only. So this whole area is an A band. M line's in the middle of the A band. The A band basically contains two different regions. It contains a region of overlap, and it contains a region of myosin only. The region of myosin only is the H zone. Line these up a little bit better. So my H zone is going to be like from there to there. always going to be found within the A band. Myosin only. No actin. The next term is the Z disc. A Z disc is kind of similar to an M line, but instead it's in the middle of the actin thin filaments. It's usually drawn as a zigzaggy disc. Again, it's a series of proteins that kind of binds all those actin thin filaments together. And that's my Z disc. And this gets me to my last vocabulary word, which is a sarcomere. Z disc to Z disc is one sarcomere. We're going to do all of this again on Tuesday because the sarcomere is the contractile unit. It is the smallest unit of contraction of a myofibril. So let's kind of talk a little bit about contraction. Contraction is getting shorter. When contraction occurs, this whole cell gets shorter. But the whole cell gets shorter because every single myofibril inside of it is getting shorter. So how do myofibrils, these are the contractile organelles, how do they get shorter? They get shorter because the actin thin filaments get pulled toward the M lines. Z disc to Z disc is going to get shorter. Sarcomeres are going to get shorter. And if that's happening for sarcomere next to sarcomere next to sarcomere, that's how this entire myofibril gets shorter. Right? And then if that's happening for this myofibril, the next one, all the myofibrils in the cell, that's how the whole cell gets shorter. So what we're going to learn about on Tuesday is how these actin thin filaments get pulled toward the M line to create or generate this contraction, right, for the shortening to occur. The filaments are going to slide. They're not growing and shrinking. We're not making new proteins. They literally are just gonna slide to get shorter and slide back to relax. Slide to the M line to get shorter, slide back to relax. That's what's happening. Sliding filaments. That's what contraction is all about. Right? Yeah, you're doing it. So elbow to elbow, sorry, one sec. Elbow to elbow, right? Super far. All I have to do is slide to get elbow to elbow shorter. Right? My arms don't have to grow. They're just sliding. That's what's happening on both sides of the M line then. Question?
Does the eye band ever like fully disappear, or is it like always present?
I'm
even, like,
so
when it
glad you
slides
asked
over?
that. Yeah. So the eye band can fully disappear during a full contraction, which is called tetany. Right? When it's fully contracted, you end up with no eye bands. Yeah. So you can imagine then during contraction what's going to happen to all of my regions here that we just defined. Okay, I just said Z disc to Z disc gets shorter, so the sarcomere gets shorter. What about the eye bands? Yes, right? This half of the eye band is going to get shorter. This half of the eye band is going to get shorter. They're going to get shorter. So eye bands get smaller, sarcomeres get smaller. That all happens during contraction. H zones get smaller, right? As the actin thin filaments are pulled toward the M line, the H zone gets smaller, smaller, smaller. But the A band stays the same length. The A band never shrinks. I know this has been a long lecture. I just just wanna show you one more thing, 'cause this is it. This model is supposed to be one sarcomere. Black line to black line is Z-disc to Z-disc. And I can move Z-disc to Z-disc. The gold bars are my I-bands, half an I-band, half an I-band, right? You have the other half over here. The red bars are my thick filaments. So ends of the red bars are the A-band. As my as my actin thin filaments get pulled toward the M line, you can observe how the H zone is getting smaller, smaller, smaller. Smaller, smaller, smaller. Right? Until the H zone disappears. The I bands can disappear. Right? In this model we know, based on the length of them. But they're getting smaller, right? So I band, gold bars only. Right? Relax, they're nice and fully elongated. As we contract, those I bands are getting smaller. So this is an opportunity. On Tuesday, I would say would be a great day to kind of play around with this model and just, define all your areas and review what's happening during contraction.
So I'm not too sure what we do to make that contract.
So that's Tuesday's lecture. I don't want to get too much into it now, but it has to do with your nervous system, right? This voluntary tissue. So we're actually going to bring in a neuron on Tuesday. We're going to talk about how that neuron tells the cell that it's time to contract and what the cell does to actually do this. Yeah. So today it's all about understanding the anatomy of the cell, the vocabulary, so that on Tuesday when I start talking myofibrils, when I start talking A-bands, I-bands, myosin and actin, you know what I'm talking about. So we're going to draw this whole thing out again on Tuesday. Okay, that's it for today. Right? This is the textbook illustration of what I just drew on the board. So you have an opportunity with a labeled illustration to review. And then we'll pick up here with some details of the filaments that are important for this to happen. So we're actually going to incorporate this last slide into Tuesday's lecture. So taking a look at the details of the myosin and the details